Okay, so welcome to this next video in which we are discussing acute gastric ulceration. Okay, so we've now discussed the anatomy of the stomach, we've discussed the histology of the stomach wall, and we've now discussed how the parietal cells of the gastric glands secrete hydrochloric acid. What we now want to look at is factors controlling the secretion of hydrochloric acid by these parietal cells. Okay, and what we'll start off with is the enterochromatin-like cells and the gastrin pathway. Okay, so basically there is a cell type that sits just underneath parietal cells. So if I get my picture of the anatomy back here, okay, so here we have the parietal cells shown in pink. Basically, just underneath these, in the lamina propria here, you'll have a special type of cell known as an enterochromatin-like cell. Okay, so let's draw this picture bigger down here. So we'll draw a parietal cell again. So here's our parietal cell with this characteristic shape where it's got these canaliculi, okay, like so, and it's sitting on a basement membrane underneath, so I'll draw the basement membrane next. So here's the basement membrane, and then underneath that, in the lamina propria, we then have this special type of cell known as an enterochromatin-like cell. I'll give it a nucleus. Okay, so here is its nucleus. So this is an enterochromatin-like cell. Okay, and enterochromatin-like cells are often abbreviated to ECL cells. So enterochromatin-like cell or an ECL cell. E for entero, C for chromatin, L for like, and then cell. Right, so what is the job of these enterochromatin-like cells? Well, basically they control the secretion of hydrochloric acid by the parietal cells, okay? And they do this by secreting a little signaling molecule known as histamine. Okay, so histamine is a very important signaling molecule. It's very important in the acute inflammatory response. It's also a neurotransmitter in the brain. It's not one of the key neurotransmitters in the brain, but there is histaminergic neurotransmission within the brain. And here's another example of its use in regulating the secretion of hydrochloric acid by these parietal cells. Okay, so these enterochromatin-like cells release a basal level of histamine, okay, so they're always secreting a little bit of histamine. And basically, the histamine is what tells these parietal cells to always secrete a little bit of hydrochloric acid. So, the amount of histamine the parietal cells secrete is directly proportional to the amount of hydrochloric acid the parietal cells secrete is directly proportional to the is proportional rather than directly proportional. Is proportional to the amount of histamine that they receive. Okay, so if they receive more histamine, they will secrete more hydrochloric acid, and if they receive less histamine, they'll secrete less hydrochloric acid. So these enterochromatin-like cells are always secreting a little bit of histamine, and therefore the parietal cells are always secreting a little bit of hydrochloric acid. Okay, so basically the parietal cells have on their surface a receptor for histamine, and this receptor is a G protein coupled receptor, and specifically it's the H2 receptor. Okay, so I'll show this here. So here are the 7 membrane-spanning alpha helices, and the amino terminus will be on the extracellular side, and the carboxylic acid terminus will be on the intracellular side. So histamine binds to this receptor, and it's an H2 receptor, so it's a histamine type 2 receptor, and basically the histamine binding will activate this H2 receptor, and it will then activate a heterotrimeric G protein, specifically the GS heterotrimeric G protein, and that then activates adenylyl cyclase enzymes which produce cyclic AMP, And the cyclic AMP then activates protein kinase A enzymes, and the protein kinase A enzymes phosphorylate and activate the proton potassium ATPase. Okay, and remember, it's this proton potassium ATPase that drives the whole secretion of hydrochloric acid. So the more activity you have in this, the more hydrochloric acid you secrete. Okay, so that's how histamine binding to these H2 receptors triggers hydrochloric acid secretion in these parietal cells. And if you want a discussion of that heterotrimeric G protein in more detail, please watch my video on gastric acid secretion. I draw that entire pathway out in that video. Okay, right. Now, basically, there are mechanisms for controlling how much histamine the enterochromatin-like cells release. Okay, so gastrin is one of the hormones which controls the secretion of histamine by the enterochromatin-like cells. So let me get another piece of paper and then we'll discuss this. Okay, so basically, gastrin is involved in increasing the hydrochloric acid secretion by the parietal cells in response to eating a proteinaceous meal. Okay, so let's show this. So, if I show the anatomy of the stomach here, so here's the esophagus going into the stomach like so, and then you'll have the pylorus down here. Okay, so this is our stomach here, and basically remember we divided the stomach into these three separate regions. We have the fundus up here, the body, I'll go to here, and then the antrum down here before you then have the pyloric sphincter. So let me colour in these different portions. So in turquoise down here, this is the antrum, turquoise. In blue, this is the body in the middle. Okay, there we go. And then in purple up here is the fundus. Okay, so basically we know that the body of the stomach is the site of hydrochloric acid secretion by the parietal cells. Okay, but the site of secretion of gastrin is the antrum of the stomach. Okay, so the G cells in the wall of the antrum of the stomach will secrete gastrin, and they'll secrete gastrin in response to protein, basically. So when you eat a proteinaceous meal, it will come down into the stomach, and some of that protein will go into the gastric pits of the antrum of the stomach, and then they'll go into the oxyntic or gastric glands, and then they'll make their way down to the G cells at the bottom of the gastric glands. Okay, and they will stimulate those G cells, and remember G cells is short for gastrin cells, and these gastrin cells will then secrete gastrin, and they secrete the gastrin into the blood. Okay, so this is going to be an endocrine form of signaling. So the gastrin then goes into the blood, and then it's going to come out of the blood at the site of the enterochromatin-like cells within the body of the stomach. Okay, so it's going to come out of the blood and stimulate the enterochromatin-like cell. So I'll draw our enterochromatin-like cell back again here. Okay, and basically it acts on a receptor on the surface of the enterochromatin cells called the cholecystokinin type 2 receptor. Okay, so this is the cholecystokinin type 2 receptor. So I write cholecystokinin at least once out in full. So cholecystokinin is another hormone released by the gut in its own right, but this receptor for cholecystokinin is also used by gastrin. Okay, so cholecystokinin. Right, so the first C is for chole, the second C is for cysto, and then the K is for kinin. Right, and then this is the type 2 receptor. So I'll colour the cholecystokinin type 2 receptor in orange. So basically gastrin will bind to that cholecystokinin type 2 receptor and will stimulate this enterochromatin-like cell, so this is an ECL cell, to secrete more histamine. Okay, so remember the enterochromatin-like cells have this basal level of histamine release, which causes a basal secretion of hydrochloric acid by the parietal cells, but they can also increase their secretion of histamine, and if they increase their secretion of histamine, then of course the parietal cells will increase their secretion of hydrochloric acid. Now, what causes the ECL cells to increase their secretion of histamine? Well, it's the binding of gastrin onto the cholecystokinin type 2 receptor. So now they're going to secrete much more histamine. So you'll get a sudden surge of histamine released by these enterochromatin-like cells, and that will trigger more hydrochloric acid secretion by the parietal cell here. Okay, so that's the mechanism by which we increase our hydrochloric acid secretion in response to eating a large meal. Okay, right. So there is another thing that regulates parietal cells, and this is input from postganglionic parasympathetic cholinergic neurons. Okay, so let me draw this here. So basically, parietal cells are also innervated by postganglionic, which means they're the second neuron, i.e. they're the neuron after the parasympathetic ganglion. Okay, so postganglionic, and then it's parasympathetic, so we're talking about the rest and digest system. Okay, and then cholinergic because they will be secreting acetylcholine. Okay, and then it's a neuron. So postganglionic parasympathetic cholinergic neurons from the vagus nerve. Okay, so cranial nerve 10. So CN10 I'll put like so. Okay, right, so CN is for cranial nerve 10. Okay, so basically these postganglionic parasympathetic cholinergic neurons which come from the vagus nerve, they will secrete acetylcholine onto these parietal cells, and basically the parietal cells have a receptor for acetylcholine on their surface, and again it's a G protein coupled receptor, and this time it's an M3 muscarinic acetylcholine receptor. So here are the 7 membrane spanning alpha helices. Here's the amino terminus, here's the carboxylic acid terminus. Acetylcholine will come in and bind here. Okay, and this is the M3 muscarinic acetylcholine receptor. Now, both of the G protein coupled receptors that we've seen today, i.e. the M3 muscarinic acetylcholine receptor and the H2 histamine receptor, they are what are known as rhodopsin family G protein coupled receptors. Okay, so G protein coupled receptors are a massive great family of proteins. There's over 800 of them in humans. Okay, if you think about all of the different species on the planet, the number that we have found is totally mind-blowing. Okay, so we've found 800 alone in humans. So to sort of, give us some sort of understanding of this, we categorize them into families, and one of the main families is the rhodopsin family of G protein coupled receptors. So the rhodopsin family of GPCRs, and this is the biggest family of the GPCRs. It contains around 750 of the 800 members of G protein coupled receptors, so it contains most of the human G protein coupled receptors. There are other families, and I'll just state these here for completion. There is the secretin family of G protein coupled receptors, there is the glutamate family of G protein coupled receptors, and I should just give a few examples of G protein coupled receptors in each of these. Secretin contains the receptors for parathyroid hormone, the receptors for glucagon. Glutamate contains the metabotropic glutamate receptors as well as the GABA B receptors. Then we have the adhesion family of G protein coupled receptors, which contains a bunch of receptors which bind to extracellular matrix components. And then finally, we also have a family named after the 2 major members, which are the frizzled receptor and the taste2 receptor. So this final receptor family is called frizzled /taste2. Okay, and as I say, it's named after 2 of its major members, which are the frizzled receptor, which is involved in the Wnt beta-catenin pathway, and then the taste 2 receptor, which is involved in the sensation of bitter compounds, the tasting of bitter compounds in the gustatory system. Okay, right. So what's important about rhodopsin family G protein coupled receptors is that they bind their ligand by residues which are within the membrane-spanning alpha helices. Okay, so the ligand binds to the membrane-spanning alpha helices, like so. So it will bind to residues that are within these 7 transmembrane domains rather than residues that are on the extracellular side, and that's what makes these rhodopsin family G protein coupled receptors all linked. So M3 and H2 will bind their ligands, which are acetylcholine and histamine respectively, in the transmembrane domain region. Okay, after that what will happen is a conformational change which will allow this receptor to interact with a heterotrimeric G protein. This time it's a GQ heterotrimeric G protein which is activated. That activates phospholipase C beta, which then cuts phosphatidylinositol 4,5-bisphosphate PIP2 into diacylglycerol and inositol 1,4,5-trisphosphate. The inositol 1,4,5-trisphosphate causes release of calcium from the intracellular stores. The diacylglycerol and then the calcium then activate protein kinase C, and protein kinase C then phosphorylates and activates the proton potassium ATPase in the same way as would protein kinase A. It's a slightly different phosphorylation site, but the principle is the same. Okay, so that triggers secretion of hydrochloric acid by these parietal cells. Okay, so not only do we have the enterochromaffin-like cells triggering secretion by these parietal cells through histamine, but we also have vagal postganglionic parasympathetic cholinergic neurons that are stimulating these parietal cells to secrete hydrochloric acid. Okay, right. So those are the major mechanisms that regulate the secretion of hydrochloric acid by the parietal cells. And I should just say that the vagus nerve will become active when we have eaten, basically. So the brain will activate the vagus nerve when we've just eaten, and therefore you will also get secretion of secretion of hydrochloric acid by parietal cells due to the vagal nerve inputs. Okay, in response to having eaten. Okay, so what we now want to turn our attention to is gastric ulceration, okay? And basically, I want to begin with Cushing's ulcers, then I want to talk about NSAID-induced gastric ulceration, and then I want to talk about treatments that are available for acute gastric ulceration. Okay, so basically, acute gastric ulceration is when you effectively burn a hole in the mucosal layer of your stomach wall. Okay, so let's get our histology back. So basically, you can have gastric ulcers that are of different severities. Okay, so you can get gastric ulcers where only the epithelial cells have been damaged. Okay, that's a very shallow gastric ulcer, a very superficial gastric ulcer, a less severe gastric ulcer. But more severely, you can get huge gastric ulcers, well, much deeper gastric ulcers where the entire mucosa is basically digested. So you have large holes in the gastric mucosa where basically it's been digested or broken down by the hydrochloric acid. So that's what acute gastric ulceration is. It's when the stomach wall, the mucosal lining of the stomach wall gets burnt by the hydrochloric acid. And two things can cause it, or potentially it's a mixture of both of them. Basically, if you're over-secreting hydrochloric acid by the parietal cells, that can lead to the hydrochloric acid being too strong, and then that will burn the stomach wall. Alternatively, if you have problems with secreting the mucus or secreting the bicarbonate onto your surface, i.e. you have problems with your defenses against the hydrochloric acid, that can also lead to acute gastric ulceration. Okay, now acute gastric ulcers are generally around 1 centimetre in diameter, and they're generally circular in form, so this sort of size, okay, and generally because of the pathological process that leads to their formation, you generally don't just have one of them, you'll generally have multiple of them. Now the good news is that once you remove whatever it is that is causing the problem, okay, whether it's intracranial injury or NSAID therapy, then basically the stomach wall can completely heal. You can regenerate the mucosal lining once it's been burnt off. Okay, so 1 centimetre in diameter, generally, they're circular, and also it's burning of the mucosa. Now it doesn't generally go underneath the mucosa, okay, but you can have differing depths. Maybe you've just got the epithelial cells damaged and the basement membrane is still intact, so let's just draw this picture, okay, so if we've got our basement membrane still intact, here's a gastric pit here with the gastric glands, like so, and if this is all still intact but the epithelial cells have been damaged, so let's say this is the basement membrane and it's just the epithelial cells which have been burnt, okay, then that would be quite a shallow or a superficial gastric ulcer. On the other hand, what you could have is large holes basically in your gastric mucosa where basically the whole thing has been burnt out by the hydrochloric acid, okay, or corrosed away. And this is a more deep gastric ulcer, but both of them can be healed. The stomach can regenerate all of this gastric mucosa that has lost. But remember, the gastric mucosa consists of the epithelium, the basement membrane, the lamina propria, and then the muscularis mucosae. So these acute gastric ulcers don't generally go beyond that. They don't get below the muscularis mucosae. Okay, right. So the first type of gastric ulcer I want to discuss is Cushing's ulcer, okay, or a Cushing ulcer. And basically, these are due to an intracranial injury, basically. So if you get some sort of intracranial injury, then you can end up with acute gastric ulceration. Now, how does this work? Well, basically, the thoughts are this, that the intracranial injury causes activation of the vagus nerve, inappropriate activation of the vagus nerve. Okay, so what happens is the postganglionic parasympathetic cholinergic neurons that are innervating the parietal cells of the stomach that come from the vagus nerve then all start releasing neurotransmitter inappropriately. Okay, so the neurons of the vagus nerve are inappropriately activated by the intracranial injury. Okay, this triggers oversecretion of hydrochloric acid, and that oversecretion of hydrochloric acid then burns the hole in the stomach wall and therefore causes the acute gastric ulcer. Okay, so that is called a Cushing ulcer, when you've got an ulcer due to an intracranial injury which causes oversecretion of hydrochloric acid. In the next video, what we'll talk about is NSAID-induced gastric ulcers.